Quantitative PCR, Session 4, Plate Preparation and Data Analysis, presented by Robert O'Brien. In Session 4, we will be going over the following topics: importance of pipetting technique with quantifiler, preparing standards, preparing reactions, avoiding and detecting contamination during qPCR setup, starting a run, analyzing data, the IPC, and using quantifiler results. Let's talk about the importance of pipetting technique with quantifiler. The standards in quantifiler are the basis by which all quantity determinations are made using the SDS software. All quantities of samples are calculated based on the curve that is made from the standards. Because of this, great care must be taken when preparing the standards. Most of the problems that are associated with quantifiler runs and the subsequent data are due to problems with the standards, which are usually because of pipetting mistakes. Now we will go over some tips for improving accuracy and precision when pipetting. Tip number 1. Pre-wet the pipette tip. Draw in and expel the sample at least 3 times before drawing in the sample for delivery. The purpose is to prevent evaporation that can occur in the tip. Pre-wetting the tip increases the humidity within the tip and therefore reduces the amount of sample evaporation. Tip number 2. Work at room temperature. Allow liquids and equipment to equilibrate to room temperature. The volume of the sample delivered by air displacement pipettes varies with air pressure, relative humidity, and vapor pressure of the liquid. All of these are affected by temperature. Therefore, working at a single constant temperature will minimize this variation. Number 3. Examine the tip before dispensing the sample. Look at the outside of the tip to see if there's any liquid present. Liquid on the outside of the tip will increase the amount of liquid being dispensed. The outside of the tip can be wiped before delivery of tip contents, but this must be done carefully, because if the material being used to wipe the tip contacts the tip opening, it can absorb the liquid inside of the tip. Number 4. Use standard mode pipetting. Choose standard mode pipetting rather than reverse mode pipetting. What is standard mode pipetting? First, push the button to the first stop of the pipette, then place pipette tip in the liquid approximately 4 mm below the surface. Slowly release button to draw up the liquid. To dispense, slowly push down to the first stop and then push all the way to the second stop. The speed of drawing up and dispensing the liquid should be the same. Reverse mode pipetting is when you push the button to the second stop to draw up the liquid and only push to the first stop to dispense. Reverse mode pipetting is used for liquids with a high viscosity. Number 5. Pause consistently after drawing up the liquid. Pause with the tip in the liquid for 1 or 2 seconds after drawing up the sample. The amount of liquid in the tip bounces slightly when the plunger stops. Slow, even plunger release and a consistent brief pause after drawing up the liquid minimizes errors resulting from the bouncing. Number 6. Pull the pipette straight out. After drawing up the sample, pull the pipette straight out. Do not allow the tip to touch the sides of the container. With very small volumes that are less than 50 microliters, surface tension effects cause the sample volumes to vary if the exit angle varies. Touching the tip against the sides results in the loss of sample. Number 7. Minimize handling the pipette tip. Avoid handling the tip and set the pipette down between sample deliveries. Body heat transferred to equipment during handling disrupts temperature equilibrium. Already stated was that temperature can affect the volume of the sample delivered. In forensic science work, the pipette tip should never be handled. Number 8. Immerse the tip to the proper depth. Immerse the tip 2 to 5 mm below the meniscus and well clear of the container walls and bottom when drawing up the sample. Inserting the tip too far into the liquid causes excess droplets of liquid to cling to the outside of the tip. Pressing or resting the tip against the container walls or bottom restricts the entry of the sample. Number 9. Use the correct pipette tip. Securely attach a tip designed for use with the pipette. Mismatching a tip and pipette or using poor quality tips can result in an inadequate seal between the pipette and the tip. Quality tips are flexible and have thin walls, providing an airtight seal and more dependable delivery of the sample. And for the final tip for better pipetting technique, number 10, use constant plunger pressure and speed. Depress and release the plunger smoothly with consistent pressure and speed for each sample. Now that you know the keys for better pipetting, let's go into how you prepare the standards. First of all, there are 8 standards that are run in duplicate on every plate. The highest concentration is 50 nanograms per microliter and the lowest concentration is 0.023 nanograms per microliter. The first standard is made with the DNA standard provided by Applied Biosystems that comes with the quantifier kit. To this standard is added TE buffer or a TE buffer glycogen mixture. Glycogen is added to the TE so the standards would last longer once made. From this initial standard and TE buffer mixture, subsequent standards are made following a dilution series. The dilution series is shown in this chart below. The H standard in the human kits contains 14 to 16 copies of a diploid single copy locus, whereas the H standard in the Y kit contains only 7 to 8 copies of a haploid single copy locus. Each well in the 96 well plate must have 23 microliters of PCR primer and reaction mix and 2 microliters of sample to give a total volume of 25 microliters. The primer/reaction mix is made up with 10.5 microliters of the quantifier human primer or quantifier Y primer depending on the assay being conducted, and 12.5 microliters of the quantifier reaction mix per well. When calculating how much of each to use, make sure to include samples, standards, and controls, and a few extra for pipetting error. Now let's go through the steps of preparing standards. First, you're going to label 8 tubes for the standard series. Vortex the quantifier human standard and then pulse spin. Prepare serial dilutions using the quantities in the table previously shown. Remember, the standards are done in duplicate for each assay. During the preparation of standards, great care must be taken when doing the dilutions. Change the pipette tip when going from one standard to the next during the dilutions. Now, let's make up the master mix, which is a combination of the reaction mix and the primer mix. Before making the master mix, swirl the reaction mix by hand. Do not vortex the reaction mix bottle. Vortex the primer mix and pulse spin. To make up the master mix, you add 10.5 microliters of primer mix and 12.5 microliters of reaction mix per well. Briefly vortex the master mix and then pulse spin. Dispense 23 microliters of master mix into each well being used. To the appropriate wells, add 2 microliters of the standards that were made, and to the other wells 2 microliters of the samples. Seal the plate with an optical adhesive cover, sealing all edges and between well spaces with the sealing tool. Remove any air bubbles that are in the sample wells by centrifuging the plate. This is necessary because bubbles in the plate can show up as small amounts of DNA when there is no DNA present. Now let's go through some steps for avoiding contamination during quantitation. Even though the 2 microliters of sample being used for quantitation will not be carried to STR amplification, it is still important to avoid contamination of one sample with another. If 2 samples are mixed during setup of the plate, then the quantities will be wrong and the incorrect amounts of sample will be taken on to the amplification process. There can be cross-contamination from one sample to another, contamination from an outside source, for example the person setting up the plate, or contamination from amplified DNA. Let's go over some ways to prevent sample-to-sample contamination. Number 1. Clearly label extraction tubes and only have one tube open at a time when loading the plate. Number 2. Arrange extraction tubes in the same order as they're going into the plate, and move the extraction tubes after the sample is loaded to a different location. This will help avoid the same sample being used twice. Number 3. Strip caps can be used to cover a row of wells on the plate after they are loaded. This prevents loading the same row of wells twice. It also prevents passing over loaded wells with a tip that contains DNA. Number 4. When loading samples, load them as close to the bottom of the well as possible, and do not depress the pipette any further after it is pulled out of the well. And number 5. Once the plate is loaded and sealed with the optical adhesive cover, do not tilt or turn the plate in such a way that would cause the samples to get onto the cover. Now let's go over some ways to prevent contamination from an outside source. Always wear your PPE while loading the plates, especially face masks and gloves. Ensure the area where the plate is being set up in has been cleaned with bleach solution before the plate is set up. Have a separate room for plate setup and where the instrument is located. The instrument is amplifying DNA, so it should be in a room dedicated for amplification. The setup should be in a room where there is not amplified DNA. Ways for detecting contamination. An optional blank can be loaded into the plate during plate setup. This is different from the extraction blank because this will specifically detect if there was any contamination introduced during plate setup or if the plate was contaminated before being used. The most common type of contamination problem with setting up the plates is well-to-well contamination, because unlike tubes, it is difficult to cap each well after it is loaded. It is important to keep an eye on your results. If a sample that you expected results gives you a negative, and the sample next to it gave extremely high results, then it is possible that a well was double loaded and the next well was skipped. Another indication of doubling of samples in a well is if a sample is run for both total human and Y, and the Y gives a much higher result than the total human, then it is possible that the Y well was double loaded. A lot of ways to detect contamination comes from thinking about results obtained from quantitation and determining if the results make sense. This will become easier with experience. Now that we have set up our plate, we can now go to the instrument. First, turn on the instrument and allow it time to start up. Open the AB Prism 7000 or 7500 SDS software and set up the plate document for the sample run. Usually there will be a template document that can be opened, so only sample names will need to be filled in. Make sure the correct template is selected. Templates may vary based on the assays being performed, whether it is total human only or human and Y combination plates. Once the sample names are filled in on the plate document, then check the thermal cycle conditions. The first step should read 95 degrees Celsius for 10 minutes, the second step 40 cycles at 95 degrees Celsius, and the third step a whole cycle for 1 minute at 60 degrees Celsius. Verify that the sample volume is set to 25 microliters and that the 9600 emulation box is checked. Save the plate document when you are finished. For the 7000, place a compression pad on top of the sealed plate, gray side down, directly over the reaction wells. For the 7500, no compression pad is needed. Place the plate in the instrument and close the door, and now start the run. Once the run is complete, the plate must be analyzed. First, verify all the analysis settings. The threshold should be set at 0.20000, the baseline start cycle should be 6, and the baseline end cycle should be 15. If any of these are different, they must be changed before analysis. If after the plate is analyzed, it is noticed these values are not correct, then they can be changed and the plate reanalyzed. To view the analysis settings before analyzing the plate, under Analysis, go to Analysis Settings, and the screen should be the same as this one below. You will see that the threshold is set and the start cycle and end cycle are clearly shown. If the plate is already analyzed, then check here for the analysis settings. Look under the Amplification Plot tab and look over to the right-hand side. If any of these are not the same as these numbers here, then they can be changed at this time and the plate reanalyzed. To analyze the plate, go to Analysis and select Analyze, or click the green arrow shown below. If anything is changed that affects analysis of the plate, or if the plate needs to be analyzed, this arrow will be green. Click on the arrow to analyze. After analysis of the plate is complete, the following should be reviewed: the standard curve, the amplification plot, and the report. The standard curve gives the following information: the R-squared value of the regression line, which shows how well the line fits with the values from the standards, the slope, which gives an indication of efficiency, and the y-intercept. To select the standard curve, under the Results tab, go to the standard curve and verify the slope for the human is between -2.9 to -3.3, and the slope for the y is between -3.0 to -3.6. The R-squared value should be greater than or equal to 0.98. This screenshot from the SDS software shows the standard curve. First you have the Results tab, under which you have the Standard Curve tab, and then to the right of the standard curve you have the information for the slope, the y-intercept, and the R-squared value. The values for the R² and slope should be in the ranges shown for the data to be used. If these values are not within range, then there may have been a problem when setting up the standards that made the standard curve. This reinforces how careful one must be when making standards, since if these values are not in range, the plate may not be able to be used and the procedure may have to be repeated. Only with the standards for a Y plate can an adjustment be made. The standard 8 or H standard for the Y kit only has 7 to 8 copies per 2 microliters of the haploid target locus. Because of stochastic effects when using such a low amount of DNA, the CT values for this concentration of DNA are more variable. This variability can throw off the line of best fit, and this in turn will throw off the R² value. So the standard 8 or H for the Y kit can be dropped by omitting the wells and reanalyzing. To omit the well, select the well, then go to the well inspector and click on Omit Well, as seen in the screenshot below. After a well is omitted, under the Plate tab, the well should be crossed out. Notice that the arrow is now green because this indicates that reanalysis of the plate is necessary because a standard has been omitted. Here are some suggestions for when troubleshooting an amplification plot. If the R² value is less than 0.98, then perhaps the quantity values entered for the quantitation standards in the well inspector incorrect, the serial dilutions were not made up properly, the standards were not loaded correctly, or the reaction of one of the standards may have failed. If the slope is not in range, then perhaps the assay was not set up correctly, the software may not have been set up correctly, the reagents may be expired or the wrong ones used, or the instrument may have malfunctioned. For the y-intercept, there is no fixed value. The y-intercept is the expected CT value for a sample that has a quantity of 1 nanogram per microliter. The y-intercept may shift from one kit lot number to the next. The y-intercept from all runs performed with a certain kit lot number should be within an acceptable range of the intercept value when the kit was quality checked. This range is lab dependent. After the plate is analyzed, then the amplification plot is checked to see what samples gave results and which were negative for DNA. The amplification plot is a plot of the delta Rn versus the cycle number. This screen shows the corresponding wells for the samples. Each individual sample can be checked to see if the sample crossed the threshold or not. Samples not crossing the threshold are negative, that means no DNA is present. Also, the IPC can be checked for all of the samples. All IPCs should cross unless inhibition occurred. This is a screenshot of the amplification plots for all the samples and the IPCs. Notice on the right-hand side under the detector, all is selected, which means both human and IPCs are being shown. The wells corresponding to the standards and samples are below, and these are in gray, which means that they have all been selected. The individual wells can be selected one at a time. Also, putting the cursor on a plot will show which sample it corresponds to. Under Detector, there is a drop-down menu to select either All, IPC, Quantifier Human, or Total Y. This screenshot shows only one sample being selected under the amplification plot. The sample is grayed out. There is a red line, and this is showing the threshold which the sample must cross to be positive, and there are two lines for every sample. One which is representing the quantifier human plot and one representing the plot of the IPC. To see either the quantifier human or IPC by itself, then go to the detector drop-down box and select what plot you would like to view. This is the same sample that was shown in the previous slide, but now only quantifier human is selected, so now you will only see one plot on the amplification plot. The report will show the quantities calculated for the samples. The IPC well has no value under quantity because no quantity calculations are made for the IPC. If more than one sample has the same name, then they are grouped together by the software and the mean value and standard deviation for the samples are calculated. During practice plates, duplicating samples is a good way to test precision of pipetting technique. Here is a screenshot showing the report screen. Under the Results tab and the Report tab, you will see the results screen. Here we have 3 samples that are selected, as you will see from the grayed out boxes. They all have the same name, and the software automatically calculated the quantities, the mean quantity, and the standard deviation of the samples. Let's talk about the IPC. The IPC stands for Internal PCR Control. The IPC is included as part of each reaction well. It undergoes the same PCR reaction as the sample, but because its target sequence does not exist in nature, it does not compete with the primers and probes that target human DNA. However, because the IPC is undergoing PCR along with the sample, anything that negatively affects the PCR of the sample will also affect the PCR of the IPC. Since the IPC is affected by problems with PCR that affect the sample, it can be used as a quality control check. The IPC can detect the following: presence of inhibitors, problems with assay setup, and chemistry or instrument failure. If the IPC crosses the threshold and the sample does not, then the sample is considered a true negative. Notice on the screenshot that all is selected, which means for that sample, both the quantifier human and the IPC should show up. Only the IPC is showing to be crossing the threshold, therefore that sample is a true negative sample. There is no DNA present. The CT range for the IPC Vic dye should be between 20 to 30. Notice on this screenshot that most of the CT values for the IPC are crossing the threshold between cycles 20 and 30. If both the human-specific target and the IPC fail to amplify, it is not possible to determine if the negative is due to no DNA present to amplify, or if it is due to something affecting the PCR reaction like inhibition. If inhibition is suspected, then the sample can be cleaned. Cleaning can be done by concentrating the sample. Some inhibitors will pass through the filters during the washes. If the sample was already concentrated, then it can be diluted. This action can reduce the concentration of the inhibitor and in turn reduce its effect on the PCR reaction. If neither of these cleaning methods work, then the sample can be re-extracted using a cleaner extraction method. Some extraction techniques like the organics extraction produces a cleaner end product. Inhibitors are usually removed during organic extractions. If this is not possible, then a new sample may be taken from the evidence that may be free of dyes or dirt or other materials that could have caused the inhibition. If the IPC does not cross the threshold and the sample has a low CT value and a high delta Rn, that means there is a lot of target DNA present. In this case, the DNA may have outcompeted the IPC for certain components necessary for amplification. Earlier it was said that the IPC and target DNA have different primers, so they did not compete with each other for those. However, there can be competition for polymerase and other components like magnesium, which is needed for PCR. If the sample has a high CT value and a low delta Rn, and the IPC did not amplify, it could mean there was partial inhibition. Depending on how low or high the quantity is, the sample may still amplify and give a result during STR amplification, or the sample may have to be cleaned up before continuing with the DNA analysis process. Now that the quantifier plate is run and the samples analyzed, what do we do with those results? First of all, let us consider if a sample that gives a negative result be taken on any further for DNA analysis. Some laboratories choose not to carry the sample any further in an attempt to conserve money and time. During quantifier, the sample is put through 40 cycles of PCR. The PCR that occurs in the next stage of DNA analysis only puts the samples through 28 cycles. Theoretically, if a sample does not amplify in 40 cycles, it will not amplify in 28 cycles. As a result, a true negative quantifier result will not be expected to generate a profile. The practice of dropping a sample after quantitation is laboratory dependent. If after quantitation there is DNA in your sample, then the extract must be taken on to STR amplification. The final value from the quantitation will be in the units nanograms per microliter. This simply means that for every 1 microliter of extract there is a certain amount of DNA. STR amplification kits require a certain amount of input DNA to give optimum results, meaning a full profile with good peak heights. Usually targeting an input of 1 nanogram of DNA for STR amplification will produce a good profile. Now let's go over some examples. If the final quantity is 1 nanogram per microliter, then by simply adding 1 microliter, one will have the necessary amount of target DNA. But what if the value is 4.25 nanograms per microliter? That means for every 1 microliter, there is 4.25 nanograms of DNA. So, 1 microliter of your extract will be greater than the optimum input level for the amplification kit. This sample needs to be diluted before it can be amplified. So let's do a simple calculation. If there are 4.25 nanograms in 1 microliter, then in 1 nanogram you'll have 1 divided by 4.25. So for 1 nanogram of DNA you will need to add 0.235 microliters. However, 0.235 microliters of a sample is too little to be added. Pipetting error is common when using such low quantities of sample. By diluting the sample, more of the diluted sample can be added and pipetting errors can be reduced. Let's look at a dilution by a factor of 10, which is simply done by 1 microliter of the extract to 9 microliters of the TE buffer. After dilution by a factor of 10, simply multiply the volume by 10. So 0.235 microliters by 10 is equal to 2.35 microliters. So, for a quantity of 4.25 nanograms per microliter, it should be diluted by a factor of 10 and 2.35 microliters of the diluted sample be added to the amplification tube. Now let's look at the other end of the scale. If at the end of the quantitation the quantity of DNA is 0.365 nanograms per microliter, that means 1 microliter of sample will not give 1 nanogram of DNA. So to calculate how much microliters will be needed to give 1 nanogram of DNA, let's do the following calculation. We have 0.365 nanograms for every 1 microliter. To get 1 nanogram, it'll be 1 divided by 0.365, and that is going to equal to 2.73 microliters. So you will need to add 2.73 microliters of your extract to get 1 nanogram of DNA for amplification. If you're using a 25 microliter reaction for the STR amplification, then the amount of sample being added is limited. If the quantity of DNA is too low, then it will require more than the amount of DNA that can be added to the reaction. In these cases, it is advisable to concentrate the DNA and quantitate again before going on to amplification. At the end of the quantifiler process, samples should be ready for STR amplification. As long as the quantifiler results show there is enough DNA that the amplification kits require to generate a full profile, then the analyst should be confident they will be able to generate a full profile at the end of STR analysis. One important limitation to consider is if there is a mixture of DNA in the sample, the quantity generated is how much total DNA is present. The resulting profiles may be 2 or more incomplete profiles or 1 full profile and 1 incomplete profile. Total human quantitation cannot tell if the final value is from more than one person. This is now the end of session 4, and it is also the end of the quantitative PCR lecture series.